Hello guys, today in this video we talk about DNA. DNA stands for deoxyribonucleic acids. Based on the definition, DNA is an important biological molecule that carries the genetic information for development and function of all cells and in many viruses. The DNA in the cell is a long-term storage of information so that the genetic information can be passed down from an adult to their offspring. In eukaryotic cell, the DNA is mainly found in the nucleus. Because the DNA is a long polymer, in order to fit the DNA within the nucleus, DNA must be tightly packaged into a specific structure called chromosome. Here we got a diagram here. We got a long polymer of the DNA, we got a histone, we got a nucleosome, and then the DNA is packaged into chromosome so that the DNA can be fit within the nucleus. Small amount of the DNA can also be found in mitochondria. Here we got a diagram of the mitochondria. Mitochondria is the powerhouse of the cell, responsible for providing energy the cell needs to function properly. If we compare the DNA in the nucleus and in the mitochondria, unlike the nuclear DNA is come from both parents, the mitochondria DNA can only be inherited from the female parent. So in the future, I will make a video and talk about why mitochondria DNA can only be inherited from the female parent. In prokaryotic cell, for example bacteria, the DNA, because the bacterial cell lack of nucleus, so the DNA can be only found in a specific region of the cytoplasm called nucleoid. For example, this is a bacterium, and then we got flagellum, we got cell wall, we got the plasma membrane, we got the ribosome, and then this one is a nucleoid where the DNA can be found. Because based on the definition, DNA is a repeating polymer. It means that DNA consists of repeating units called nucleotide. Each nucleotide contains one nucleobase, one phosphate group, and one deoxyribose sugar. Here we got the diagram. We got the phosphate group here, we got the deoxyribose sugar, and we got a nucleobase. The nucleobase can be adenine, guanine, thymine, and cytosine. The example here is an adenine. Here we got a structure of the DNA. We got adenine, we got guanine, we got thymine, and we got cytosine. Adenine and guanine are purine, thymine and cytosine are pyrimidine. If we look at the structure here, adenine and guanine contain 2 carbon rings, while thymine and cytosine contain 1 carbon ring. So this is the reason why thymine and cytosine are pyrimidine, adenine and guanine are purine. For the formation of the DNA, it follows a specific rule called Watson-Crick base pairing. Adenine and thymine fit together as do those of guanine and cytosine. Based on the base pairing rules, adenine always pair with thymine, guanine always pair with cytosine. If we look at the diagram here, adenine pair with thymine with 2 hydrogen bonds, and guanine pair with cytosine with 3 hydrogen bonds. The hydrogen bonds in the DNA is important because the hydrogen bonds help to stabilize the structure of the DNA and form the double helix. 2 nucleotides are connected by a covalent bond called phosphodiester bond, which is a linkage between the 3' hydroxyl group of one nucleotide and the 5' oxygen of the next. The DNA strand in the double helix has directionality. If we look at the diagram here, this strand is run from 5' direction to 3' direction, while the complementary strand is run from 3' direction to 5' direction. Well, the 3' and the 5' is refers to the sugar itself. If we simply number this carbon atom of the deoxyribose sugar, the first carbon atom, second carbon atom, and the third carbon atom. So here we got 3' N. if we number the carbon atom here, we got the first carbon atom, second carbon atom, third carbon atom, fourth and fifth carbon atom. So this is the reason why we got the 5' end here. If we look at the structure here, the 3' end is terminated by a hydroxyl group, and the 5' end of the DNA is terminated by a phosphate group. Well, here we got DNA double helix. If we look at it, the sugar phosphate backbone is pointing toward outside of the helix, while the base pair point toward inside of the helix. It is because the sugar phosphate backbone is highly hydrophilic. Hydrophilic simply means water-loving, so by pointing toward outside of the helix, it allows the backbone to interact with the water molecule. The base pair point toward inside of the helix because the base pair here is highly hydrophobic. Hydrophobic simply means water-fearing. Here we got the structure of the difference between the ribose sugar and the deoxyribose sugar. If we look at it, the second carbon atom is attached to a hydroxyl group, while the second carbon atom of the deoxyribose sugar is attached to a hydrogen atom. So the ribose sugar, we got the hydroxyl group here, and the deoxyribose sugar, we got the hydrogen atom here. Based on the stability, DNA is much more stable than RNA. It is because the hydroxyl group here can act as a nucleophile that donates an electron to the electrophile. There are two forces help to stabilize the structure of the DNA. The first one is the hydrogen bonds between the nucleobases, and the second one is a base stacking interaction among aromatic nucleobases. So that's the end of the video. Thank you for watching. Don't forget to like, comment, and subscribe to our channel. Any questions, comment below or email us. Thank you.